Okay, welcome everyone. It's got a nice robust turnout today. That's always good. Today is our regular Thursday conference, and sometimes we have various student presentations. And one of the steps along the way to the PhD degree is a symposium where a student kind of gives the background and sets the stage for their dissertation work. And so we're going to hear that today from Leung, who's a PhD candidate in our program in the BCB track. Eric's a graduate of University of Oklahoma and received dual bachelor's degrees in math and biochemistry. And while he was there, he helped assemble the genome of the Anopheles gambiae mosquito, which is carrier of the malaria parasite. He also worked in a lab studying weakly electric fish and trying to sequence a potassium channel protein. He became interested in biomedical informatics when learning about a risk assessment making use of a patient wellness portal. That's bioinformatics? No. But that's great. It is. It very much is. Some days patients will look up their genome in their patient portal. Anyways, he also learned about the Health Information Exchange Network that was being developed in Oklahoma. And during his time here, he's transitioned to learning about bacteria in your gut and how it influences health, which we actually heard about yesterday too, the microbiome. And he's interested in applying computational methods to understand these interactions. He's also well prepared for it because he's one of the few BCB students who has taken BMI 510. Anyways, I won't delay any further Eric's talk.
Can you hear me? Okay. All right. Hi, everyone. Thanks for coming to my talk. So today I'll be talking, as Bill said, about the area of research that I'd like to go into. And what it's called is the Microbiota-Gut-Brain axis. And specifically, I'm interested in its connection with mental health. And so I'll start off talking a little bit about some history on each of these three components and how they eventually converged into being studied together. And then I'll go into some case studies in which they were actually studied together, and then go off into talking about some potential areas in which I could go into. Let's see. Maybe not. Okay. Okay. So just briefly on the microbiome. I'll get into it in more detail in just a bit. But for now, I would just like you to know that the microbiome is just a collection of microorganisms that exist in and on you, as gross as that may sound. But they are the collection of the microbes in your body. So what I like to do is propose a question for you to think about during my presentation. So the microbiome, or specifically the ones in your gut, can they influence your brain and even your behavior? And even if they can have some sort of influence, how can these three characters, the gut, the bacteria in your gut, and also your brain, how can they even talk to each other or have some sort of relationship, kind of like in this cartoon behind me? So out of the three, let's start with the brain, and specifically in mental health. And so there are a variety of mental health disorders, and two of which I want to focus on are depression and anxiety. And so both of these are quite prevalent globally. And so according to the World Health Organization, over 300 million people around the world suffer from depression. In terms of anxiety, here in the US, it's estimated about 18% of the adult population has anxiety. Not only is there a societal burden on that, there's also a financial burden with the high prevalence of that. So there's some estimates that say for anxiety in the US, it costs about $42 billion, and depression costs upwards around $210 billion a year. So with these numbers are quite staggering and make it important or a motivation to study these sort of diseases. So yeah, with these staggering numbers of its prevalence, and also, or these numbers show the high prevalence and the financial burden of these diseases. Not to mention the also possible personal and social burdens that we might face in our lifetimes as well. So all of which can contribute to why we might want to study depression and anxiety. So we started on the brain, and now let's incorporate the gut. And so people have been studying the brain and the gut together as early as the ancient Greeks. And so one example is this guy over here called Galen of Pergamon. And so he was a physician, a philosopher, and surgeon. And he was well known for being an expert in human anatomy. And so one thing he observed is a physical connection between the gut and your brain in terms of the nerves being innervated throughout your gut and also various other organs. Fast forward to the late 1800s, we come to the development of more conceptual or a theoretical framework to think about the connection between the gut and the brain. And so two people I want to highlight are these men over here. And we have Carl Lange, who is a Danish physician, and we have William James, an American physician. And so surprisingly enough, they were actually able to independently think of a theory of emotion, and which is now known as the James Lange theory of emotion. And emotion being anywhere something like love or fear and anger. And so what this theory says is that we feel emotion as our brain's reaction to initial physiological responses. So say, I guess, a more gut-related phenomena is like, say you're in love, and the typical thing that you may feel is, butterflies in your stomach. So what their theory would say is that you would first have some gut reaction, and then your brain would perceive that, and then you would feel this emotion of love. So even though there are more modern theories of emotion, or the connection between your brain and other organs, this theory laid the groundwork for a lot of them that exist today. So years go by, and researchers continue to dive into this interaction between the brain and the gut. And so they discovered this bidirectional signaling or communication that the brain and gut have. And so bidirectional means that there's a top-down signaling from the gut to the brain, and also a bottom-up signaling from the gut to the brain. So in the next few slides, I'd like to just highlight briefly those directions independently. So from a more top-down view, going from the brain to the gut, two major mechanisms in which the brain can influence the gut are the autonomic nervous system and the HPA axis. And so with focusing on the autonomic nervous system, this nervous system is involved in controlling involuntary actions of your internal organs, such as your heart beating or digestion. And so even within the autonomic nervous system, there are two pieces to it, the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system is related to what is known as the fight or flight response, or something that if you're stressed, your body will react in that way. Additionally, there's the parasympathetic nervous system, which is associated with what's called the rest and digest response. So your organs will do different things in these different states. Going into the HPA axis, which stands for the hypothalamus pituitary adrenal axis. And so what this axis is responsible for is our body's ability to control and respond to stress. And so how this one might be related to how it influences the gut is that these three organs might release some sort of molecules, which will then piggyback on the autonomic nervous system and send certain signals down to the gut. Now going the opposite way, going from the gut to the brain, we have three major mechanisms for this as well. We have a more immune response, an endocrine response, and a mechanical or neural response as well. So an immune response in our gut is another barrier between the outside world and our internal organs. So you can imagine the amount of immune cells that might need to be there to protect from any pathogens that might enter our body. And so the immune cells there might be able to interact and send signals back up to the brain. Additionally, endocrine molecules such as serotonin and histamines might be generated in the gut, which also will affect the brain as well. And lastly, there could be a mechanical feedback from just your digestive tract trying to get rid of certain foods, and also, the foods that are there to move it along. So that kind of feedback loop will also get to your brain. And so now we come to a point where all three, you include now the microbiota in there, and we get the microbiota-gut-brain axis. And so this addition, or this discovery of all three together has made an impact, and you could even say there's been a paradigm shift in certain fields like neuroscience and psychiatry. And the reason being is that even without the microbiota, those disciplines have long ignored or haven't really taken account even the long amount of research that has been happening in the gut and brain connections that I've mentioned before. And so now with the advent of new technology, we can now understand or at least measure some of the bacteria in our gut, and that has an additional piece that can be added to the mix. So this allows a way for you to at least further analyze or understand these highly complex diseases and neurological disorders. And so much momentum has been gained that even some funding or some organizations worldwide have become interested in this topic, such as the NIH here in the US and also in the EU. So I mentioned briefly about the microbiota, and here I've come to talk about it again. So the gut microbiota specifically are the trillions of bacteria that live inside of your gut. And so two large groups of them, if you remember briefly in our taxonomy classification, if we look at the genus level, which is pretty low, the largest group is called the Bacteroides. And then if you go a little bit higher up in the taxonomy and looking at the phylum level, the most that we have are the Firmicutes. So in my talk, I'll be focusing on the gut microbiota's interaction with the brain specifically, but just in general, a little more information about the gut microbiota. It's also implicated in various other diseases as well, such as obesity and diabetes, and several other gastrointestinal disorders as well. So there's been a thought that the connection between your gut microbiota and these diseases is because of some imbalance or some differences and changes in your gut, also called, or what people call dysbiosis in your microbiota, that may have some influence on these diseases in some way. So now I'd like to switch gears into talking a little bit about some case studies that involve all three, the microbiota, the gut, and the brain, into studying how they're related to some mental, or specifically anxiety and depression. So we'll talk about an initial study in which trying to link depression and anxiety with the risk of a gut-related disease. Then I'll go into some behavioral experiments in rats and mice, and how they decided to infer or suggest that the microbiome might be involved in anxiety. And then I'll end with another case study and focusing on depression in humans as well. So to start off, there's this one study. It was conducted because in 2000 there was an unfortunate contamination of some drinking water in a Canadian town, and which led to an outbreak of pathogenic E. coli. And with this E. coli, it spread it started to spread essentially the stomach flu around, and which had serious consequences. And so at the time, people have known that having the stomach flu is a risk factor for developing something called irritable bowel syndrome, or IBS for short. And so at the time, the pathogenesis of IBS was poorly understood. So what these researchers wanted to do is take this opportunity to study this prospectively. And so they did so following these individuals in this town for a total of 8 years. And so just to show that there is a link or some sort of association between the stomach flu and IBS, here's a plot showing the association between the individuals who have either self-reported that they have the stomach flu or that have been clinically diagnosed in the more darker bars. And then you have unexposed controls from the contaminated water in the white bars down here. And so as you can see, the effect of having the stomach flu increases your prevalence of IBS. And so again, I mentioned that IBS at the time, people didn't understand the pathology quite so well. And so they took this opportunity to then try to find some risk factors for IBS. And so some risk factors that they decided to look at were age, female or gender, anxiety, and then certain other acute illnesses or features of acute illnesses. And so what they did is a multiple logistic regression, and the results of that analysis are shown in this table. I don't want you to take in this table just yet, but the results of this is certain odds ratios for each of these variables. And an odds ratio generally is a quantification of the strength between certain these variables and some other variable of interest. In this case, it's the incidence of IBS. And so the one that I want to focus on here is prior anxiety and depression. And so in each of their years, they ran this model and tried to see if there's some association. And so in the earlier years, there wasn't any significance for prior depression to be a risk factor for IBS. But then in 2008, their model showed that there is a statistical significance between the two. And so this implies or suggests that there might be some connection between depression or some mental health issue and gut-related issues. And so for the next couple case studies, they moved to more animal models for studying anxiety. And so two of the tests that I want to highlight here are called the step-down test and the light-dark box test. And so for the step-down test, you have a box, and then you have an elevated platform in the middle. And so what you do is you'll put the animal on top of that, pedestal or platform, and then you'll wait to see how long it would take for the animal to step down from that platform. And so this is a measure of anxiety, because if you're in a new area, you might not want to explore it as much. So the more anxious animals might just stay on there a little bit longer and take quite a bit of time to get down, while the more exploratory ones and less anxious animals might be quicker to come down and explore the area. A similar test is the light-dark box test, in which you have two compartments, a more lit area and a darker area. And again, this is a test for anxiety, in which less anxious mice or animals will want to explore and be in the lit area, while the more anxious ones will stay in the darker areas as well. So you can measure the time in which they're in the light area as an indication of how anxious a certain animal is. And so again, so there's a study that made use of these two tests. And so what they wanted to do is try to look at the influence of the microbiota and certain behavioral and brain chemistry changes. So here they have a model in which they had control mice, which is just a normal developing mice with all its microbes. And then you have the same kind of mice, but then they subjected them to a cocktail of antibiotics or antimicrobials for a whole week to try to really disrupt all the bacteria that they might have in their gut system. And so then they subjected them to these anxiety tests. And so on the left, you can see the results from their step-down test. And so the controls, they took about a little over 200 seconds to walk down from that platform. However, the antibiotic treated mice, they took only 100 seconds to get down. So this suggests that they're less anxious and they're able to explore, or they would want to explore a little bit more. Again, suggesting maybe that the microbiota might have some influence on anxiety. Similarly, in the box model, the box test, you have the controls to see how long they would be in the lit area. And so for the controls, they were in there for about 150 seconds, while the antibiotic-treated ones stayed in the light areas a little bit more, about 225 seconds or so. So again, similar results in that, or a different way to test similar results, that the microbiome might be influencing our behavior and making us less anxious. What's more is they decide to take these antibiotic-treated mice and then let them recolonize their bacteria normally after 2 weeks. And so then they subjected them to the same test and to see how they would do. And those results are in these grayish bars over here. And as you can see, they have partially or almost completely restored the behavior to what would be considered normal for these mice. So again, suggesting that there might be some interaction between our gut microbiota and behavioral changes. So another concept that I'd like to bring up that might help with the next case is a concept of germ-free mice and specific pathogen-free mice. And so germ-free mice, as you might guess, are mice that, or animals that don't have any germs or bacteria. So they have to live in a very sterile environment and eat sterile food. Meanwhile, for the specific pathogen-free mice or animals, these guys would have specific bacteria which they would be screened for, so that at least there would be a limited or somewhat known set of bacteria in which they don't have, which can help for certain studies. Additionally, another concept that will come up is this concept of a microbiota gut transplant, or you might have heard as a microbiota fecal transplant. And so what this will do is a procedure in which you'll take the fecal or stool, result samples from an individual, and then you'll transfer it into another person or animal. And you can do this either orally with some pills, or you can do it the other way with an enema. And so in this experiment, they wanted to make use of the step-down anxiety test. And what they wanted to do here, which is really cool, is they wanted to make use of the behavioral differences in 2 strains of mice. Specifically the bowel mice, which is known to be a little bit more timid and more anxious than the other mice, which is the NIH Swiss mice. And so here we have where they had each of these strains being germ-free, and then subjected them to this anxiety test. Then they had another group of these same mice, but then had the microbiota of the other strain of mice being transplanted into them, and then see how well they performed in these anxiety tests. And so focusing on the left part, we have the NIH Swiss mice. And so as mentioned, that the NIH Swiss mice, they are a little bit more exploratory and less timid than the bowel mice. So their time to step down from this pedestal or this platform is about 20 seconds. Meanwhile, when you incorporate the microbiota from the other strain, the time in which it takes them to step down from this platform increases to about 60 seconds. Similarly, in the bowel mice, they would take they're again known to be more timid and more anxious. So, as expected, with the normal, I guess, SPF and the germ-free mice, they would take quite a bit of time to step down, of about 280 seconds, which is quite a bit more than the NIH Swiss mice. However, when you subjected them to a fecal transplant with the other strain, the time in which they took to step down also, decreased. So again, these results show that, or indicate possibly that the gut microbiota can also influence our behavior and anxiety, and that it can be transferable. So additionally, not only in behavior, this group wanted to look at brain chemistry differences. And so focusing specifically on the NIH Swiss mice, after they transplanted the microbiota of the other strain, they waited 3 weeks, and then they measured the BDNF levels in the hippocampus and the amygdala. And BDNF is an acronym for brain-derived neurotropic factor, and is known to be a neuroprotective and neurogenesis factor in the brain. And so when you subject them to the other kind of microbiota from the different strain, you can see some differences, at least in the hippocampus, in terms of how much BDNF factor is being generated. Of course, in the amygdala, you can see here that there wasn't any change. So even though there wasn't any change in the amygdala, there was at least some measurable difference in the hippocampus, suggesting not only that the microbiota might have some behavioral influences on the rat or mice, but also a chemical change as well. So in another study, they did a similar experiment looking at at least one of their experiments, they wanted to look at the light-dark box test as well. And this time comparing the germ-free and specific pathogen-free mice. And so from here, you can see that the specific pathogen-free mice want to spend more time in the dark areas. So they were a little more timid or more anxious. However, once you get rid of their microbiota for the germ-free mice, you can see that their time in the light area increased significantly. Again, suggesting that there might be some connection between the gut microbiota and our behavior. So another test they did was called the open field test of anxiety. So again, we have a box, but this time there's no platform or anything. So now, what you want to concentrate on is either the center or the open area, and then the walled area. And so this again is a test for anxiety, because the mice might want to, or the animals might want to, stay more closely to the walls rather than the open areas. So for this test, you want to see how willing these animals are to spend their time in this unknown open area. So what this group did is they wanted to look at a possible developmental dependence of the gut microbiota. And so what they have are 3 groups of mice, So they have germ-free mice, they have a specific pathogen-free mice, and then they have what's called a conventional mice, in which you start off having a germ-free mice, and then you add in a normal bacteria colony later. And so they did it in early life and also in adult life. And what I have here are the results from when they did it in early life. So they had mice grow up to a certain age in early life, and then they recolonized them to be specific pathogen-free. And so I mentioned that this is using the open field test, and one way to quantify their anxiety is what's called the number of rears that they have, or the number of times in which they stand on their hind legs, which is an indication that they might be more open to the idea to explore things and less anxious. And so I want to point out here 2 the white dots here and then the black dots. So the white, or the specific pathogen-free, are the ones that have some sort of bacteria in them, while the black dots are the ones that are germ-free. And so the gray dots here are the ones that are conventionalized in young, early life. And so as you can see that after you conventionalize them a little bit, it partially restores their behavior towards what would be considered normal in this case. Again, suggesting that there is some influence on the microbiota and even developmental changes in our brain. So again, they did this also in adult life. So these mice were grown up to be germ-free until their adult life, and then they were conventionalized with some microbiota, or some bacteria. So unfortunately here we see that there is no change between the ones that are germ-free and the ones that were colonized in adulthood. So again, both of these suggest that there is some window in which that our microbiome might be important in correct brain development and normal behavior. So the last study that I'll focus on is turning into more depression. So in the previous studies, we're focusing on anxiety. And so what this group did is, firstly, they wanted to look at what the compositional differences in the gut microbiota in patients with depression and healthy controls. And to look at compositional changes, there's two metrics at least to look at, which are called richness and diversity. And so richness is simply just the number of different species or groups in the sample, while diversity takes into account not only the number of different species, but also their abundances as well. And so comparing both the controls to their depression counterparts, you can see that the depression patients had less number of different groups, and also less diversity in their guts. So again, suggesting that at least in depression, there's also some change that happens that might be associated with depression. So not only that, so they focus solely on humans first, but then they want to try out if they can even replicate that depressive phenotype into rats as well. And so for rats, they started off with normal rats, and then so they raise them into a certain age so they can develop normally with bacteria. And then they'll subject them to 4 weeks of antibiotics, trying to wipe out their entire microbiome, or their microbiota. And then they'll subject them to a fecal transplant from those depressed patients. And then they'll try some behavioral tests to see if they can recapitulate those kind of depressive phenotypes in these rats. And so one test of depression in animal models is to see their preference to sucrose. And so, excuse me. So when both of them, the control mice and the fecally transplanted mice, were subjected to this sucrose preference test, the depressive mice now prefer the sucrose less so than their control counterparts. So suggesting that there is a transfer of this depressive phenotype to not like certain things anymore, similar to in maybe a human. Additionally, they did an open field test, and again, they had this box. It was just a wide open field, and the places of interest are the center, open area, and then the walls. And so here, they found that the controls would be in the center for about 15 to 20 seconds. Meanwhile, the depressed mice, and now I guess you can consider depressed mice or rats, they only stay in the center for about 5 seconds. So again, trying to relay the behavioral phenotype into these rats, suggesting a strong influence on the gut microbiota in depression. So a summary of these cases so far is that we've seen a connection between a gut-related disorder, IBS, and anxiety and depression. So some connection there, along with the contamination of the drinking water. And then we looked at some studies that try to recapitulate or look at the effects of the microbiota in anxiety and development. And lastly, we just covered going into depression and how it's related or possibly suggests to be associated with depression and changes in the microbiota. So now I'd like to switch into some areas that might be interesting to go in from here. Namely, some network methods, some neuroimaging, looking at multiple body sites, and then I'll end with talking about the concept of hallmarks of this axis. And so talking about the ecological and network methods of studying the microbiome, or the microbiota. So scientists so far have maybe looked at the microbiota in a more empirical sense, where they would add in maybe one or two or a couple of these certain bacteria so that they can study it in animal models. However, as we know that the microbiota has so many different species that you can't keep on doing this empirically. And at some point you have to look at all of them together and all of these interactions together in possibly a network model. And this is powerful in order to capture all of these different interactions that might happen between a group or a certain subset of the bacteria. Additionally, there might be some network signatures, once you take into account all of these bacteria, some network signatures that might help in predicting certain diseases. So there's this one group that tried to make use of the microbiota in a more clinically diagnose, or to clinically diagnose Crohn's disease. And so they wanted to see if adding in new information about the network of these interactions of the bacteria can better diagnose or predict Crohn's disease better than just clinical features alone. And so they were able to find that and give some hope for network models to help predict disease. Additionally, you can go for a more ecology method and looking at the microbiota over time and how it deals with certain changes or perturbations to it. And so one example of how the microbiota might initially become stable and be okay, and then fall into some sort of unstableness or a disease state is Clostridium difficile, C. diff, in other words. And so this introduction into your gut can wreak havoc on your gut system and provides an example of this kind of model where initially your gut is normal and then some change, the C. diff is introduced, and there's a great change in instability in which it propagates to the whole digestive tract. And so network models and using ecology is a very powerful method to studying these interactions in the gut microbiome. So another method that could be used is neuroimaging. So I touched briefly on the chemical differences that the microbiome might add to or change in our brains. So additional measurement you can do is to image the brain itself to see what it's up to. And so one example of this is called resting state functional MRI, in which an individual or the animal organism would be in just a resting state, not doing much, and it would be subjected to an MRI, which would look at the changes in blood flow in the brain. And so an example of at least the neuroimaging part being studied in some gut disease. Here's an example in which they want to look at indigestion and possible changes in the brain. And so what they want to do is take healthy controls and people with indigestion and try to use machine learning algorithm to try to differentiate the two. And so even though this model doesn't use the microbiota in per se, one can speculate that adding in the microbiota could also give you some extra information to increase the amount of predictability power that you have in distinguishing between healthy controls and some disease state. So another area in which you can make use of the microbiota is looking at multiple body sites or different microbiotas. And so here I have an example of just the diversity that can be found on the skin. And so you can see the compositional differences shown in these small pie charts. And you can see that they all vary across the body site based on maybe some dry areas versus some more oily areas. And so one example of a group making use of this, again, not focusing on the gut or the brain, but in this case they looked at pregnancy. They looked at different parts of the different microbiota, such as the fecal matter, the oral cavity of both the adult and the infant. And even comparing all those, you can see, so this is a principal components analysis plot in which it tries to find, separate differences or be able to plot them in a 3D space and to visually see that each of these parts have a distinct area or sort of signature to them. And so what you might be able to do is, along with the network methods, is try to incorporate all or multiple microbiotas to try to get a larger picture of how maybe a certain disease state is affecting not only just one ecosystem, but another as well. And so I want to wrap up with talking about this interesting paper suggesting a sort of hallmarks of the microbiota-gut-brain axis. And so I'll briefly go over them: the indistinguishability, emergent properties, bidirectionality signaling, a critical development window, and neural homeostasis. Sorry. So indistinguishability is the concept of that from now on we can't really study the human without considering our microbe counterparts. And so this was suggested in the Human Microbiome Project that started in 2007, in which they suggested that humans should be studied as a supraorganism, which again is you have to study the human part, but also with our microbe friends as well. And again, it's suggested in some of the cases I showed that even in development or even our normal behavior can be affected by our microbiota changes. So again, thinking about them being as one, and you should be able to study them together. So the concept of emergence is sort of where I was heading with the network methods, in which you can't just study maybe one or two of these bacteria by themselves, but however, when they all come together, they come up with certain emergent properties that might not otherwise be seen in these individual bacteria themselves. And in terms of bidirectional signaling, again, I touched on at least the bidirectional signaling in the gut and the brain, but you can also add in the microorganisms in there as well, because they would also interact with the immune system, or just as a review, some of the interactions between the gut and the brain are the immune, endocrine, and neural connections. So for the immune system, it has to deal with what it considers to be foreign, and the bacteria are still considered non-human. So they will have to regularly interact with our microbiota and be able to send signals to the brain. Additionally, we have endocrine signals such as serotonin and histamines, which can be developed or be byproducts of some certain metabolism of the bacteria in our guts. And lastly, the neural part of it is the bacteria just being on our gut might send some signals to our brain. And there are certain bacteria such as segmented filamentous bacteria, in which they adhere to the gut itself. And so that can also signal to the brain as well as some certain things going on. Critical window of fluidity, I mentioned this briefly in one of the cases in trying to look at the dependence of the microbiota in development. And so this says that, again, there's some window possibly that if we don't develop with our microbiota, there could be some long-term effects in terms of behavior and brain development. And this also makes sense from an immunological standpoint as well, because as we grow up, or from being an infant, you're exposed to all these different microbes, and you have to be able to distinguish which ones are good and bad. And if you aren't exposed to these harmless ones, then if you're exposed to them later, your body will think that there's they might be pathogenic, and you'll have some overreaction to them. And lastly, I want to touch on neural homeostasis. And again, touched on the BDNF factor, which is involved in neural protection and just like keeping a healthy brain. And so, again, the microbiome might also be implicated in keeping a healthy and normal brain state and behavior. So that covers the hallmarks of the microbiota-gut-brain axis, and that concludes my presentation. So thanks so much, and I'll take some questions. Yes.
So you brought up a lot of, for the hallmarks, a lot of different approaches that could be taken. What I'm trying to determine is where you see, because this is a symposium, and one of the things we can do is kind of give you feedback as you're trying to think about your proposal.
Sure.
Where is the low-hanging fruit that you're really, because what I want to avoid is kind of that laundry list of here's things that could be done. Where do you is the low-hanging fruit, especially something that might actually really have an implication for an intervention or implications for health?
Mm-hmm. Yeah, so in the depression case I saw, or that I shared, they did a multitude of other tests as well. So they not only looked at the diversity and richness that I mentioned, but they also looked at cytokines and stuff like that. But their analysis, they only looked at maybe certain differences in certain microbes. So they didn't take into account maybe certain like low abundance microbes that might influence those more highly abundant ones. So I feel like maybe more network methods could be a low-hanging fruit in that case. So, yes, Ted.
This is a question that probably
doesn't have an answer.
It might not have an answer right now.
Okay.
Like, it's something to think about. So how would you identify like an emergent property, like compared to like just like the individual contributions?
Mm-hmm. Yeah. So I haven't studied emergent properties intensely, but I would guess you could try to find, if you had be able to look at the microbes themselves, maybe get a subset of them that might be highly characterized, and try to, I guess one property that you could think about is their metabolic output, so the byproducts in which they have in terms of just them living. And if you have, say, focus on some subset, smaller subset, and you can know what they output, and maybe in when you put them all together, you could also measure their output and see if maybe, a very naive way, but see if their output is what it would be expected if they were all just individual and independent from each other. And so you might be able to compare those differences, but I'm sure there are more sophisticated methods to identify emergent properties. Yes?
Yeah, this may actually be a little rephrasing of Shannon's question. I understand, and the biology is really interesting. Mm-hmm. I'm trying to get kind of get an understanding of what aspects of bioinformatics would really address some of these problems critically. Yeah. So what kinds of tools or techniques? Really, what kinds of questions are you going to address that would be amenable to the kinds of approaches we apply to various kinds of data that would come out of this?
Mm-hmm. So one way would be kind of like the example I showed in trying to differentiate between like a healthy state and a disease state using certain machine learning algorithms. And so that would make, they would take into more of a sophisticated view in kind of differentiating what kind of bacteria are in one state or another. Because maybe a naive way to look at it is maybe look at maybe the top 5 bacteria that might be in each of them and try to compare those. And that might not be enough. So you could use to make use of all of them and to see if there's also maybe some differences between the two to help with, I guess, either diagnosis or prediction of this disease. So, yes?
So it kind of sounds like the idea that you're extracting here is similar to what you might find in environmental microbiology, and that keystone species aren't always the most abundant species.
Right.
So are there particular techniques that you want to use to, so bioinformatic techniques or related, that you want to get at those rare species?
I mean, that's an area that could look into. One way would be to maybe, because sometimes you might not be able to measure those rare microbes in there, so similar to those rare gene expression in like humans. And so maybe you could possibly impute the data is one way to look at it, from if you knew a priori that they might be there from other studies. So that might be a way to get at trying to get at those low abundant ones, but that's...
And then maybe more interestingly, what is it that they're doing? And how do you start to answer that question? Because there might be maybe some metabolite in low abundance, but it's critical for the functioning of the rest of the community, or certain members of the community to do their job. So to me, those are the most interesting questions. Is what are the metabolites that are being exchanged between the host and the commensal microbiota? And I don't know if you were going to be able to get at those questions. It would be interesting.
Right, to look at those low abundance ones, because that's also a possibility that the low abundant ones might be the most important ones. Again, similar to like gene expression. So, Amanda.
Just a couple of questions. One is more of a curiosity question regarding the biological background. I mean, has anyone done a study in clinically depressed patients with fecal transplants?
I do not know.
Was there improvement in depressive symptoms?
Oh, more of a intervention to do? Oh, okay. I do not know right now, but could look. So that paper on depression, that was published last year. So, I mean, within that year, there might have been.
It seems like the next most obvious step. And then secondly is much more of a practical question. I mean, are you aware of, or do you have a plan for the data sets that we're going to have access to in your project? Are you going to use public data sets? And if so, what are the data sets that are available in regards to these particular hallmarks?
So in terms of data, I was planning on maybe making use of Lisa's data, and so she has a cohort of individuals that have some sort of anxiety or depression, and also some samples from their stool. So, but in terms of the other kind of data sets I might be able to look at, I'm still collecting and seeing what else there is out there. Cool. No other questions? All right. Well, thanks so much for stopping by.